I am Mattia Giovannini, a pediatric specialist from Italy and the IACI Union member representative for the Allergy Immunotherapy Interest Group. Today, we have the pleasure to welcome Dr. Michihiro Naito, Nagoya University of Arts and Sciences, Japan, to discuss together a paper recently published on PAI journal entitled Evaluation of Cross-Reactivity between casein components using inhibition assay and in silico analysis. Thank you, Michihiro.
Thank you.
Could you please explain the rationale of this study and what are the main outcomes?
Thank you very much for this great opportunity to have an interview. The main outcome is that alpha S1 casein and beta casein showed almost perfect cross-reactivity. We have published a letter in the PAI journal, which confirms that specific IgE levels to purified alpha S1 casein and beta casein showed strong correlations in the patients with cow's milk allergy. However, this was somewhat surprising finding because the protein database shows that alpha S1 and beta casein share only 4% homology in the amino acid sequences. Generally, those proteins with such a low homology cannot cross-react with each other. But anyway, we tried to perform the ELISA inhibition assay to detect the cross-reaction. We used beta casein as an inhibitor to the ELISA system to detect alpha S1 casein specific IgE antibodies. The result shows that alpha S1 casein specific IgE antibody was almost completely inhibited by beta casein.
Thank you. The methodologies of your study are really interesting. So, what are the methodological challenges of this study?
We have 2 points. First, I could obtain highly purified alpha S1 and beta casein using many steps of fractionation. And we have strictly checked the absence of contamination in each fraction. I had a confidence about the purity of the sample, so I could believe the fact that these 2 caseins do have the cross-antigenicity. Second point, we used in silico analysis named STAP tool to find the common epitopes. A good point of this tool is that partial homology can be detected in the whole length of a protein. I set the sequential peptide consisting of 10 amino acids in the primary sequence of alpha S1 casein and applied them to the sequence of beta casein. In this analysis, we succeeded to find the amino acid sequences that is homologous between alpha S1 and beta casein. As a result, there was a well-known casein-specific sequence called as casein phosphopeptide, CPP.
Perfect. And to summarize, what is the overall conclusion from the paper?
Actually, there are 4 CPP motifs in the casein components. 1 CPP motif is present in alpha S1 casein, 2 in alpha S2, and 1 in beta casein. They have a core sequence of 3 serines and 2 glutamic acid, SSSEE, and the serine residues are strongly phosphorylated. And there are relatively conserved amino acid sequence besides the core sequence. So we do estimate that there are the major IgE epitopes in these CPP regions. However, previous report using the synthetic overlapping peptide did not find IgE epitopes in the CPP regions. We need to overcome these discrepancies. So we predict that phosphorylation of the serine residues in the CPP motif has a significant role in the antigenicity of caseins. Currently, we are investigating the effect of dephosphorylation on the IgE binding ability of casein and CPP. Hopefully, we can publish the next paper in these issues. Thank you.
Great. Thank you very much for being with us today, Michihiro, and thank you all for watching. If you want to read the full article, please click on the link below.